want to transition into some of the biological approaches that currently have an enormous influence on this field. Let's start with genetics. You know a fair amount about genetics, probably from high school biology, and we're not going to go into that just yet. Our genes do not determine who we are, but they influence who we will become. And in psychopathology, we refer to that genetic influence as a diathesis. So, for example, our current model for schizophrenia, which by the way is shared for most disorders, is there seems to be a fairly strong genetic influence that makes people higher risk for the development of schizophrenia. The influence does not guarantee that they will develop schizophrenia, but it changes something. It has to be biological, because that's all genes affect, that in exchange affects the way the person reacts to the world, and under the right circumstances, they may develop the disorder. The genetic influence is called a diathesis, but by itself, it's not sufficient to trigger the disorder. It needs another factor called the stressor. So the diathesis stress model, which we're going to be talking about throughout the entire course, is the current way that we view all forms of psychopathology. Now, how does that diathesis affect us? Well, when we're talking about psychopathology, the primary thing that we're talking about is the brain. The brain is responsible for all of our behavior and our feelings and our unconscious processes as well. The diathesis probably affects the way the brain develops and the way the brain functions. So we're interested in the neurological mechanisms, the functioning of the brain, and how that contributes to psychopathology. Now, how does the brain get affected by the genes? Well, genes are templates for chemicals, specifically proteins. And every single cell of your body, including every single neuron in your brain, is a little chemical factory. The chemicals that are produced, the number of chemicals, which chemicals are in each cell, will affect how that particular cell responds and how it interacts with the nearby cells. So genes affect chemicals, usually within the cell, which in turn affects the way the cell functions. And when the cells are part of the brain, it affects the way the brain functions. Now, none of these issues are up for debate. The evidence for them is overwhelming. Unfortunately, these are so complicated that there's a lot of blank spaces in our information and a lot of research that needs to be done. But I can tell you, having been in this field for about 45 years, that quite frankly, we have gone from ground zero to an incredible level. And that level is accelerating. So in a matter of 10 or 20 years, we may be able to tell you a lot more about how these variables function. Let's introduce you to some important concepts in genetics. Some of these terms you're probably familiar with, others less so. You may remember from high school biology that the genotype are the specific genes at a specific location on a chromosome. In the human body, we have 23 pairs of genes. And the pairs are roughly the same, with the exception of the sex genes. In males, the X gene and the Y gene are very different in size and have very different genes on them. A female has 2 X genes, and a male has 1 X and 1 Y gene. But the other 22 pairs have essentially the same gene location and can have either the same or a different gene at that particular location. So when we talk about the genotype, we're talking about what are the two genes at this specific location on the pair of chromosomes. Phenotype is the observed trait. It is sometimes related to the genotype and sometimes not. You may remember learning about Mendel's peas. Gregor Mendel was the person who, with a series of clever studies, mostly on garden peas and other plants, was able to discern that there were a pair of genes that accounted for the wide variety of different types, phenotypes. I don't remember all of the details, so I may be wrong on the details here, but the principles will be correct. Mendel found, for example, that some genes were dominant, which meant that if you had one copy of them, you would get the phenotype associated with it. Some genes were recessive. If you had one copy of it, you wouldn't see the phenotype. But if you had two copies, you would see the phenotype. So, for example, if green versus yellow, if green was the dominant, this is the color of the pea, was the dominant type, a genotype that had one yellow and one green, or a genotype that had two greens would be green in color. The only yellow peas were the peas that happened to have two yellow peas. Now, a term you almost certainly have not been exposed to is epigenetics. Epigenetics is the study of gene expression. Okay, we have somewhere in the neighborhood of 125,000 genes in the human genome. And every single one of our cells has 125,000 genes. But which genes function depend on the situation. Genes, for example, that are important in neurotransmission, that is, brain cells, will be active if the cell happens to be in the brain. Genes that are important in stressful situations may be activated by signals that the body gives that are influenced by external experiences. It says this is a stressful situation, and suddenly the chemistry of the cell changes, and therefore the behavior of the body. We are now at the level, especially when we're talking about behavior, of looking at epigenetics and trying to understand that process. You may be aware that we have now matched the entire human genome. So we know where the genes are. We know what they're made up of. And in time, that's going to give us an incredible insight into the movement from genes eventually to behavior. Right now, we're not there. Right now, most behaviors that are abnormal, that is, most psychological disorders, appear to be affected not by a single gene, but by a combination of genes. And we're nowhere near sophisticated enough to know which genes they are or how that process works. What we can do is we can establish that genes have an influence. If genes influence behavior, the behaviors will run in families. So, for example, if genes contribute to schizophrenia, then we should expect schizophrenia to run in families. Now, that doesn't mean that everyone in the family will have the disorder, but there will be more people in the family with the disorder than you generally see in families. But the problem is, if something runs in families, it might run in families because of the genes, which are shared by families, or because of the environment, because families grow up together, and it may be the environment. We need to separate these two effects, and we do it in two ways. One is the twin study. What we do is we find individuals with a disorder. If we're studying schizophrenia, we'll find individuals who have schizophrenia. And we will determine whether or not they have a twin. If they have schizophrenia and they have a twin, we find that twin and determine, one, is the twin identical? Do they look alike? Do they share 100% of their genes? Or is the twin fraternal? They're no different than siblings, except that they were fertilized, the eggs, at the same time, and the birth occurred at the same time. Identical twins share 100% of the genes. Fraternal twins share 50% of the genes. But both of them are carried together in the womb, so they experience the same environment there. They're born at the same time, so they grow up together and they experience the same environment. Now, identical twins will always be the same sex, but fraternal twins half the time will be opposite sex. And of course, boys and girls get treated differently. So the idea that, well, they grew up together, therefore they experience the same things, is not necessarily going to be true if the fraternal twins are opposite sex. So we exclude any fraternal twins who are not the same sex. That way we're controlling environment. We take a look at the twin also to find out if they have the disorder. We're studying schizophrenia. Do they qualify for the diagnosis of schizophrenia? If they do have that diagnosis, we say they're concordant. If they don't, the co-twin, we say they are discordant. The concordance rate for schizophrenia for identical twins is about 50%, just under 50%. The concordance rate for fraternal twins, same sex, is about 17%. Now, what I'm doing is averaging dozens of studies that have been done over several decades. So clearly, the concordance rate is much higher for monozygotic twins, suggesting that genes play a large role. But if genes played the only role in whether you develop schizophrenia, the concordance rate for monozygotic twins would have to be 100%. And it clearly is not. It's only about half that rate. So the genetic study with twins indicates very clearly genes play an important role, but environment does as well. Now, adoption studies take a different approach to separate environment from genes. If you're adopted, you have two families. One you know well because they raised you and they provided your environment. And one you probably know very little about. They are your biological family. They share your genes, but you didn't grow up with them, so you didn't share their environment. Adoption studies show very clearly that genes play the more powerful role in determining who will develop schizophrenia. We could spend weeks, whole semesters in fact, talking about nothing but the central nervous system and its role in psychopathology, but we don't have the time, and unless you had the background, this would be very difficult for you to follow. So I'm going to focus on the most important elements. When we talk about the nervous system, we make a distinction between the central nervous system, which is the brain and the lower extension of the brain called the spinal cord, and the peripheral nervous system. The central nervous system controls most of our thinking, a lot of our feelings. The peripheral nervous system interacts with the rest of the body for the brain to control that part of the body. Some of those connections are with organs and muscles. We call that the somatic nervous system. We're not going to spend much time on that. That's a fascinating area, but that's 2 or 3 graduate level courses alone. But we are going to focus on the other part of the peripheral nervous system, which is called the autonomic nervous system. The term autonomic is used because it is thought to be largely automatic, and in fact it is. Now, we once thought it was completely automatic, but we've been able to show in the laboratory that it is possible for people to learn how to control some aspects of the autonomic nervous system. The autonomic nervous system has two branches. The sympathetic branch is active when you are under stress. The parasympathetic nervous system is active when you tend not to be under stress. These two branches are mutually exclusive. Well, not quite, but they're close. They tend to inhibit one another. So if you're under stress, sympathetic activation dominates. If you're not under stress, parasympathetic nervous system dominates. The sympathetic nervous system gives you more energy to deal with the stress. So you might say to yourself, geez, why shouldn't I have that all the time? Then I'd always be able to handle things. Well, the parasympathetic nervous system is important because it does the housekeeping chores of the body. It's what keeps you healthy. It digests food and removes waste so that your body is healthy and therefore able to function under times of demand. The brain is probably the most important organ of the body. It averages about 4 or 5 pounds. It fills about half of our skull, but it accounts for all of our thoughts and feelings, and it basically controls virtually everything within our body. It consists of specialized nerve cells called neurons. It's estimated that we have between 10 and 100 billion neurons. And on average, the neurons in the brain are connected to about 1,000 other neurons. You can do the math. That means you have between 10 and 100 trillion connections within the brain. Now, the brain is modular. It has different parts that work semi-independently but are able to coordinate. So we can look at various parts of the brain. If you look on the left here, you will see what is called the brain stem. The brain stem has most of the critical elements for life. If damage is done to the brain stem, very likely you will die. So things like regulating your heart and breathing and things of that sort are sorted at the brain stem. The thalamus, which is part of the brain stem, is a switching station so that signals coming from various parts of the brain are shunted to the places they need to go for the brain to function. Above the brain stem is what we call the midbrain structures. It's mostly the upper part, yellow on the right side of the photo here. The midbrain structures tend to have most of our emotions and our drives, and they influence our behavior in some very important ways. In human beings, the largest part of the brain by far is the cerebral cortex, which is so large that it covers all of the rest of the brain. You can barely see the brain stem and a part called the cerebellum, which I'll talk about in just a minute. The cerebral cortex is where we think. It's where we do most of our perception. It's where we drive our behaviors and record our memories, etc. The cerebellum, which I'll come back to much later in the course, is very important because it tends to smooth out our behaviors. So instead of our hand moving in kind of a jerky way, our hand moves smoothly because of the influence of the cerebellum. But the cerebellum also seems to be involved in some forms of psychopathology, which we'll see later in the course. The lower or hindbrain is important for basic functions, and there's only one that's going to be relevant to this course. Of course, we need the organism to be alive, which means the heart has to beat. But as it turns out, this is the oldest part of the brain from an evolutionary perspective, and it is where the fear response, we often call it the fight or flight response, is triggered. We'll come back to that when we cover panic disorder in a later chapter. The midbrain structures are going to play a role in that they involve many of our emotions. But by far, we're going to focus mostly on the cerebral cortex, because all of the disorders that we talk about include disruption in the ability to think, remember, and behave, all of which are associated with the cerebral cortex. Let's talk about the nerve cells called neurons. This deserves at least 2 hours. You're going to get about 2 minutes, so it's going to be the basics. The neuron is no different than any other cell in the body, except that its operation is different. The expression of the genes in the neuron causes the neuron to transmit signals to other neurons or to other organs of the body. The signals come from branch-like or start at branch-like structures off of the cell body that we call dendrites. The dendrites might be attached to many, many different neurons. And then the signal goes down a tail-like structure that we call the axon to the end of that tail, the axon, to what's called the axon terminal. So the signal is always one way. The signal is very rapid within the cell, and without going into the details, it's an electrochemical action. The potential difference inside and outside of the cell changes because of movements of ions in and outside of the cell. And again, we're not going to talk about that. At the end of the axon terminal, everything changes, because the signal doesn't go directly to the next neuron or 100 different neurons, depending upon how many connections there might be. Instead, there's a gap. That gap is called the synapse, which we'll talk about in just a minute. This slide summarizes what I just described. Within a cell, we have an electrochemical process that goes from the dendrite down the axon to the axon terminal. Between the cells is a purely chemical process. Within the synapse, which is the gap between the axon terminal and the dendrite of the next cell, chemicals called neurotransmitters are used to send the signal. Now, this process is very slow compared to the process within the cells. Now, when I say very slow, I'm talking about a few milliseconds instead of fractions of a millisecond, but it's fairly slow. These neurochemicals, neurotransmitters, there are about 3 dozen that have been studied extensively, and probably half a dozen that we're going to learn about extensively, because they play a role in many of the disorders that we'll talk about in this course. The neurotransmitters are chemicals, just like all of the other chemicals. The template for those chemicals are in our DNA. And whether they're produced in a particular neuron depends on where that neuron is. Because remember, gene expression is not always the same in every cell. So the cell produces these chemicals. It takes a lot of energy to produce neurochemicals, neurotransmitters. Therefore, when the chemicals are released at the axon terminal, they're allowed to float for a while, where they may or may not connect with a receptor, that is a chemical that is sensitive to the neurotransmitter and will bind to it. If they do not bind, what will happen is the chemicals will be brought back into the synapse, into the transmitting cell and stored and reused. That's very important because it takes so much energy to be able to produce these chemicals that you want to be able to use them hundreds and thousands of times before they are finally discarded. That process is called reuptake, and it turns out to be very important in the field of psychopathology. The synapse doesn't look anything like this drawing. This just shows you, illustrates how it is that these things work. The chemicals, the neurotransmitters, are shown as shapes. And those shapes will bind to certain other chemicals that are in the dendrite of the next cell if the shapes match up, if they're comparable to one another. And that binding process is what signals the next cell. The neurotransmitters are stored in the cell and then released when the cell fires. There are also drugs that can have an effect on how much of the neurotransmitters are released. Those often have their effect within the synapse as well. So, for example, there's an illustration of a drug called Prozac, which we're going to talk about a lot in this course, that binds to the axon terminal and influences the process of reuptake. I mentioned the autonomic nervous system as part of the entire nervous system. It's a branch of the peripheral nervous system. The autonomic nervous system is not covered in this chapter. It's covered in a later chapter, but I want to cover it here because I think it's important to cover it here. I've already mentioned it has two branches, the sympathetic and the parasympathetic. They tend to be mutually inhibitory. And the sympathetic is active when you're stressed, and the parasympathetic is active when you're not stressed. When you're not stressed, the focus is on keeping your body healthy, digesting food, removing waste, things of that sort. Both of these divisions regulate most of the organs of your body, but usually in opposite directions. So, for example, when you're stressed out, your body temperature goes up, your muscle tension increases, your heart rate increases, and that prepares you to work harder, to deal with whatever the stress is. All of those things slow down under parasympathetic nervous system activity. There is one other biological system that's very important in the field of psychopathology. It's not covered in this chapter, although it is covered in a later chapter, but I want to introduce it here. It's called the endocrine system, and it works entirely different than the nervous system. In the nervous system, you have very rapid signals passed by neurons through the brain or through parts of the body to influence the functioning of the body. The endocrine system uses chemicals, but the chemicals are dumped into your bloodstream. The chemicals are produced, we call them hormones, in glands that produce specific hormones. And they are released depending upon the situation that you are in. If you are in a stressful situation, for example, a variety of stress hormones will be released. You're going to discover that one of the most important of them is called cortisol, and we'll talk about that much later in a later chapter. This release is coordinated through what we call the HPA axes: hypothalamic, pituitary, adrenal cortical. Adrenal cortical is what releases the cortisol and some other hormones that are part of the stress response. These are all brain mechanisms and other organs that work together. They're often coordinated by the sympathetic branch of the autonomic nervous system. The endocrine system, unlike the autonomic nervous system, is very slow to react because the chemicals have to be dumped into the bloodstream and then circulated. And that takes about 60 to 90 seconds for those chemicals to move throughout the entire body. But they have an advantage over the autonomic nervous system. because they have a longer lasting effect. If you're under a real stressful situation, the autonomic nervous system will kick your body into high gear. But it'll also trigger the endocrine system, which will kick your body into a higher gear and a more long-lasting gear, so that you will be able to do whatever it is you need to survive for a longer period of time. So the endocrine system is slower to respond, and it's slower to shut off. Now let me give you two examples that will help you to remember that concept. You perhaps, if you're a driver, have had an experience like this. You're driving down the highway, minding your own business. Some idiot isn't paying attention, and they all but run you off the road by changing lanes without signaling. And they didn't even look. They have no idea you're there. You slam on the brakes. You steer very carefully, just avoid losing control, and then bring it back onto the road. Take a deep breath and keep driving. All of that was activated by your central nervous system. It saw the danger, and it controlled your muscles to be able to control the automobile and prevent the accident. But it's dangerous, and so your central nervous system detected the danger and triggered the autonomic nervous system. So the sympathetic branch went into high gear, turned the entire body ready to fight for whatever needed to happen. And because it looked like it could be life-threatening, the endocrine system was triggered. And all these hormones were dumped into your system. But you're not going to feel those hormones for about 60 seconds or so. You responded so quickly that you took a potentially life-threatening situation and took the danger out of it. You're back on the road driving. And then 60 seconds later, suddenly you're hit with this enormous emotional response. Now, depending on your personality, that emotional response might be something like, oh my God, I almost died. Or it might be, God damn it, I'm just going to kill him. Okay, again, your personality shapes how you feel the emotion that is triggered by the endocrine system. But it seems out of place because it's 60 seconds too late. But it takes 60 seconds for that system to have its effect. The other example that will help you to remember the slow response, if you're working late into the night, or if you're under a lot of stress before you go to bed, you can't wind down. Now, it doesn't have to be negative. You could be out on one of the best dates of your life, and you're so excited, and you get home and you can't sleep because all you're doing is thinking about how great that night was. There's a tremendous amount of hormones that are circulating in your system because you're so excited, positive or negative, and it's going to take an hour or two for you to wind down. There are several treatments based on biological manipulation. We're going to talk about them in detail as we're going through the various disorders, so I'm only going to introduce them now. Medications are the most common by far, and we have effective medications for many of the disorders that we'll be talking about. Psychosurgery was much more common 50 or 60 years ago, much less common now. In fact, it's fairly rare now. Electroshock therapy was very common 60, 70 years ago, but it tends to be used very little now, although you'll discover when we cover mood disorders that it actually works, and works rather well. But since drugs often work and are a little less dramatic, we tend not to use shock therapy. Transcranial magnetic stimulation is also used for treating mood disorders, specifically depression. And unlike shock therapy, it doesn't cause grand mal seizures, and therefore is much less dramatic. It is used now, but not extensively, but it may be used more in the future. Our final perspective is actually two perspectives. One is the humanistic, and the other is the existential perspective. The humanistic and existential perspectives were very common in the 1960s into the early 1970s, so when I was an undergraduate and the start of graduate school, and it had influence in that it moved away from the sort of animalistic models that represented behaviorism and psychodynamic theory. The humanistic perspective basically argues that people are basically good, and that when pathology develops, it's because the natural maturation processes have been interfered with. If we give a human being a comfortable and safe environment, they essentially will heal themselves. The existential perspective suggests that the individual has to take responsibility for themselves. If they do not, they don't feel good about themselves, and psychopathology may result. Now, both of these are interesting theories. Both of these are things that people should know about because they do represent human functioning. But their contributions to psychopathology have been minimal for two reasons. First, neither one supports research. Both of them argue that each individual is so different from every other individual that it's unrealistic to try to study common processes. Well, that's garbage. Think about that. We're all different from one another, but people who study heart functioning learn things that help you to survive a heart attack. Because our heart functioning, even though each of us is individualistic, is remarkably similar in one person to the next. The fact that we are different does not mean that we do not share common processes, and you can do research on these processes. So this chapter covered the perspectives that have had the most influence on our field. The psychodynamic perspective at one point dominated, but it no longer has the kind of impact that it once was, largely because it doesn't have the rigorous scientific research needed to form the basis of a scientific understanding of psychopathology. In contrast, the behavioral, and now also the cognitive behavioral, which we'll talk about later, and the biological approaches, including genetics, have had a much bigger impact because they have a much stronger scientific base. The humanistic and existential perspectives, I think, influence the way we think about people. They certainly have had influence on therapists. But their perspectives on the development of psychopathology were at best simplistic, and at worst, simply myths. We now know that the processes that they talk about don't seem to occur, or at least don't have nearly the impact of behavioral or biological processes.